Hi everyone, it's Mr. Byers again. So some things are easier for me just to talk about than to necessarily show just in a PowerPoint. So I want to go over Punnett squares first. So when we're using Punnett squares, keep in mind as you read through chapter 11 that we're dealing with just one gene, two alleles of one gene. So in this example in the PowerPoint, we're talking about how in pea plants yellow seeds are dominant to green seeds, but they come in both forms. So there are yellow alleles and green alleles. Some pea plants will have yellow, some will have green. Now I tell you that the yellow seeds are dominant. So when we're writing our Punnett square, we would use the capital Y. The reason we do that is the yellow allele, yellow starts with Y, dominant, or I'm sorry, the capital letter tells us it's dominant. There's no rule saying we have to use Y. You can use any letter. That it's just sort of note-taking shorthand, but why would you confuse yourself? If you use a capital Y, that tells you, oh, yellow is the dominant one. When we write the recessive allele, we're going to use the same letter. So in this case, we have yellow and green. We're not going to use Y and G. That doesn't tell us anything. Capital Y and lowercase y tells us dominant and recessive without having to flip back through our notes. And if someone else were to look at it, they would pretty much know what was going on. That's just how you use Punnett squares. Now, Punnett squares gives us potential offspring. So we have a square. Usually it's 4 squares, but to make it on the PowerPoint, I just had to make a table like this. On one side, you line up one parent's potential alleles. So every parent has 2 alleles for that gene, for that trait, right? They could be homozygous dominant, homozygous recessive, or heterozygous, but no matter what, you have 2 alleles for that trait. The one that we see is the dominant one. So in this case, we said that the individual was true breeding, so they're homozygous dominant, and the green-seeded parent was homozygous recessive. So we would put those on either side of the Punnett square. And just as an aside, there's no such thing as heterozygous dominant or heterozygous recessive. That doesn't make any sense. Heterozygous just means one dominant, one recessive. So we line it up on either side. I put a line over my lowercase letter just because some letters look the same when you write them. So we put all the potential sperm alleles on one side, the potential egg alleles on the other, and then we cross them in the middle. We simply line them up like this. So from this parent, we got the lowercase y. From this parent, we got the capital Y. Now, all this is telling us is potential offspring. It doesn't mean that when we combine 2 pea plants, we're going to get 4 babies that are heterozygous. That's not what that means. This just gives us the potential that we will see. So we have 4 squares here. So really we're dealing with 25%, 25, 25, 25, 25 equals 100%, or 25 cents, sorry, 4 quarters and a dollar. So in this case, if we had one parent that was homozygous recessive and one parent that's homozygous dominant, the only possible children we could end up with, the possible offspring we could end up with, would be heterozygous. So their genotype would be capital Y, lowercase y, or we would say their genotype is heterozygous. So what's their phenotype? What's the physical appearance? If this lowercase y is green and this capital Y, the dominant allele, is yellow, if an individual is heterozygous, what are they going to look like? Yellow or green? Hopefully you're saying yellow, because this dominant allele masks the recessive. Actually, as we're going to learn in chapter 12 and 13, it turns off the recessive allele. We don't even make these proteins. The only proteins that are going to be translated are the yellow proteins. So we can use these Punnett squares to do test crosses. Maybe you know one parent, or you know what's recessive or what's dominant, and you want to figure out the offspring, even though you may not know the genotype. Because just looking at an organism, unless they were expressing a recessive phenotype, you wouldn't necessarily know if they were heterozygous or homozygous dominant if they express the homozygous or the dominant trait. You would have no way of knowing unless you knew the parents. So a test cross helps us figure that out. So as an example, here I give you a test cross with a short-haired cat and a long-haired cat. Long hair in cats is recessive. And so if you see a cat with long hair, you know it has 2 copies of that recessive allele. Because if it had any copy of the capital of the dominant allele, it would be short-haired. So we know when someone tells you that the individual is expressing a long-haired trait, you know it's homozygous recessive. It couldn't be anything else. But if it's crossed with a short-haired dominant, we don't know. We wouldn't know whether it was homozygous dominant or heterozygous. All we know is it at least has 1 copy of a dominant allele. So we can perform a test cross. But what that means is we can look at the offspring and sort of work our way backwards and say, okay, if we see this offspring, we know that one of the parents should at least have this allele. So like I said, we lined up one parent on this side because it was homozygous recessive. That didn't change. The short-haired cat, all we knew is it has at least 1 copy of that dominant allele. So then we can look at the offspring. If all of the offspring are short-haired, we can assume, well, let me just show you. This S would come from the one we knew about. If the offspring were short-haired, all that this parent could give would be the lowercase s. So in order for this to be a heterozygote, the dominant allele had to have come from the other parent. So if all of the offspring are, and even this is just potential, right? It's not 100%, but for the most part, if all we ever see is short-haired, we can assume that that parent was homozygous dominant. However, if in that litter of kittens we saw long-haired and short-haired, we would know that that lowercase s had to have come from this parent, so they were heterozygous. You'll want to work through a few of these yourself to see if you can answer these questions. So what I've done is I've given you a situation here, and I want you to try to figure out what the genotype and phenotypes are. Well, I'll give you the phenotypes, but the genotypes of these parents. So I give you a situation with a bull with no horns crossed with 2 different horned cows. The first cross only produces horned calves. The second cross only produces horned or polled calves. So what I've done is I've given you your Punnett square boxes. I told you what the offspring were. Now it's up to you to figure out what the parents are. If you can do this, you're guaranteed to be able to answer those questions on the test. Some topics just need more explanation, and that's why I'm here. So reach out. Send an email to kbyers@mineralarea.edu. I can help give study tips, we can set up a phone call, or even a video conference.